Okay. So we started talking about transcription. And we talked about transcription in eukaryotic cells. And the process is extremely complicated to initiate, and it's regulated very, very tightly. There are several factors involved. In order to transcribe genes, we talk about having a gene in DNA and the polymerase transcribes it and make the RNA and make the protein. But it's much more tightly regulated like that. There are several things that have to happen. The first thing is that promoters on genes have to be recognized. And there are specific sequences, consensus sequences, that genes that code for proteins have that are used for the regulation, for the initiation of transcription. We talked about one of these, the top box, right, which is in the promoter region of eukaryotic cells. In the proximal promoter, which covers about 200 or 250 nucleotides or so, there are four major conserved sequences that serve as for binding and initiation of transcription. And one of those is the top box. The other is called the GC box. Genes that are transcribed, that are housekeeping genes that are needed for a cell to survive, generally have this GC box there, which ensures that those genes are expressed in all cells. Cat box, oct box, both of those are used in different genes for different expression. We're not going to talk about that. But these specific sequences are recognized and bound by proteins, right? So proteins bind to those things and recognize them. Even the top box is recognized by protein binding. The proteins that bind to that top box, right, are called transcription factors. And so the transcription factors bind, and form a complex around that box. And in doing so, that recruits the polymerase to that site, and it also aligns the polymerase. And the protein binding there starts and goes in a sequence. This is a table that lists all these things. The sequence begins when TF2D, transcription factor 2D, right, binds to that promoter region, to the top box. And the reason it does is because that protein, the top binding protein, recognizes it. The top binding protein serves several functions. One is it recognizes the box, but also when it binds, the protein itself is a monomeric protein, but it has two identical domains. And when those domains bind to this top box region like this, right, it's like almost like a saddle sitting down on that DNA. And what it does is it bends that DNA. And we talked before about what that bending of DNA does. In this particular cartoon, I don't know if it's from your book, but it shows you pretty well what that bending does. If you look at the DNA strand, you can see the nice double helix. And if you take that helix and put that bend in it, a 90-degree bend essentially, you can see you disrupt that helix, that formation of the helix. Also, you notice right in this TATA, T-A-T-A-A-T, that TATA box has all those TAs, only 2 hydrogen bonds per nucleotide pair. And essentially, once you put that stress on there, it helps to bind and open that bubble. See now? Second thing that it does, so it recognizes that. It binds and it bends the DNA and begins to open the bubble, the transcription bubble, and then it recruits other enzymes. So here's the same sort of a picture where it's bent. You can see that nice structure there. So the TATA box binding protein binds to the box, causes that bend. When 2D binds, it recruits other proteins. It recruits 2B. 2B comes in, binds, and stabilizes the complex. When they're there, they 2B stabilizes the complex, but it also recruits 2 other proteins, 2E and 2F. And 2E and 2F are bound in solution to the 2 polymerase, the polymerase 2 molecule. So when they're recruited to the complex, they pull polymerase into the complex. So now you can see we're building this complex with transcription factors and RNA polymerase 2. Other than just bringing these 2 in there, E and F also recruit the last 2 polymerases, E and A, or F recruits the last guy, E and H. H is that one that has all those activities. You can see in this cartoon, they give you the 6 little circles. It's a helicase. So it takes that region where the TATA boxes has that stress, And the helicase uses ATP and opens it up so there's actually a bubble there. Transcription can begin. The polymerase can start working on that template now. The second thing is, remember we said that this complex, you can see here, it interacts very tightly with that promoter region, 10 to the minus 8 molar, right, in prokaryotic cells. In eukaryotic cells, it's similar, but it binds very, very tightly. So when transcription begins, this whole complex is interacting so tightly with these sequences in the DNA, right, and with the other proteins that we don't even see in this complex, right? Upstream from the Tata box, we have that CAT box, the GC box. So there's a lot of proteins in this complex. So this thing can't pull away. And what happens is that this TFH also has the kinase activity. And it's a kinase that phosphorylates serines and threonine groups, right? Here's a picture of the molecule. So here we are with everything there. We show now that what happens is we get this region here, which is called the carboxy terminal domain. It's on the carboxyl end of the protein. This protein is called the beta prime protein. That's the catalytic subunit of this polymerase. Beta prime subunit. It has this tail on it called the CTD. In order to initiate, that tail has to be totally free of phosphates. But once it does, and H is in the process, is in the complex, it begins to phosphorylate that tail. And when it does, that tail changes conformation so that it no longer interacts with the other transcription factors, with the other proteins such as OCT1 and the GC binding protein, ISP1. It doesn't interact with those. Everything gets loose again. So it's all loosey-goosey, and some of these other transcription factors fall off, and now the polymerase can continue on. You can see the only transcription factor that stays there is this TF2D complex, which is the very first one. So that when polymerase pulls away again, it doesn't have to go through this process of finding, once again, finding a promoter element, a Tata box on a gene. So it just stays there, and that way that gene can be transcribed more rapidly the second time around. You can see that this carboxy terminal domain has 27 heptad repeats, heptad, 7 amino acids. There's the repeat starting with a tyrosine. It has 4 serines and threonines. 3 of those, and I don't remember which of them, can be, I think 3 of them can be phosphorylated. So there's 108 total serines and threonines there. So you can phosphorylate this tail tremendously. 1 phosphate is enough to change the conformation of a protein. When you put all of those on there, it drastically changes it. Okay? So the first thing is to locate the promoter region of a gene and to pull in the polymerase, recruit polymerase to set up that transcription bubble. Now, DNA in eukaryotic cells is not always accessible to these proteins. It can be in various states that make it accessible or not. Whether or not these proteins bind determines whether or not a gene is expressed or not. We have 22,000 genes or so in our genome. At any one time, there's only a couple thousand, a few thousand of them that are being expressed. Others were needed for development and shut off permanently. Others are needed for tissue-specific cells. They're only expressed in muscles and not others, in blood and not others. So they're not all expressed. And whether or not they are, is whether or not these proteins are synthesized, the proteins that bind, right, like the OCT1 protein and SP1 protein in a particular cell, and whether or not those proteins have access to the promoter. Why do they have access to the promoter? Or don't? Well, remember, our DNA is all bound up in this structure called nucleosomes. You've probably seen a picture, if you read the text, you've seen a string of beads, right? Where you have nucleosomes linked together by about 50 nucleotides or base pairs of DNA. So and it's very regular. It looks like a string of beads. That string of beads is even coiled, super coiled into this is 100 angstrom across, it'll be a 300 angstrom fiber, even bigger. Okay? Under normal conditions, this DNA is wrapped very tightly around that histone core. The histone core has 8 individual protein molecules, 2 each of the 4 histones that are labeled there. In fact, it's wrapped so tightly around that proteins that interact with DNA, those DNA binding proteins, the Tata binding protein, OCT1, SP1, all of those polymerase don't have access. They can't interact with the DNA because it's so tightly bound around that nucleosome. Okay? So in order to be able to transcribe those, that DNA has to be loosened up. And the major way that that DNA is loosened up is by interactions between the DNA itself, the sugar phosphate backbone of the DNA, and the proteins. And one of the major ways that they interact is through these lysine residues and arginine residues, these charged residues in the tails, and these are on the amino terminal tails, right? That's amino acid 1, so that's the amino terminal. Carboxy would be the last proteins are made, amino to carboxy, right? So this is the amino terminal end. So the amino terminal tails of these histone molecules have concentrated or a large number of charged amino acids, lysine-rich and arginine-rich amino peptides. So, as we said yesterday, those lysine residues can be modified. If they're acetylated, they lose their charge, they become neutral. If they lose their positive charge, they interact less tightly with this histone, they loosen up. The DNA breathes, it lets loose of the histone. If they are deacetylated, then it comes back together, they tighten back up. So acetylation is a regulatory mechanism to activate genes. Deacetylation deactivates them, right? Because it allows the DNA to interact more tightly. So in order for the proteins to be able to interact, in other words, a promoter might be in this region here, tightly wrapped around that nucleosome core. In order for all those proteins to interact, this chromosome has to breathe. The enzymes that do this are called HAT. I don't think I have it written anywhere. So HAT